Okay, so this review session today, we're going to go over DNA repair mechanisms as well as DNA replication. So DNA replication. And I have a video clip that I'd like to play real quick just to go through it. And hopefully you guys can hear the audio for it. I'll make it full screen. Can people see the video clip? Just need someone so they can see it. Can you see it?
I can't see it. I'm still seeing the review session.
I'm so sorry. I did not play it. Can you see it now? Okay. So yeah, so I didn't, so sorry. So what I was saying before is that we're going to recap DNA replication. And so to do that, I have a video here. So you can all see the screen, right? I'm going to assume now you can see it.
When DNA replicates, the enzyme helicase separates the DNA strands. Single-stranded DNA binding proteins then stabilize the unwound template DNA, keeping it in an extended single-stranded state so it can be copied by DNA polymerase. One new DNA strand is called the leading strand, which forms from its 5' to its 3' end by the action of DNA polymerase III. A sliding clamp forms a ring around the DNA and maintains the association of the DNA and the polymerase, allowing the uninterrupted synthesis of many thousands of nucleotides of DNA. The machinery of the replication fork includes helicase and a primase enzyme. Primase creates short RNA segments called primers on one of the template DNA strands. The RNA primers allow DNA polymerase to initiate DNA synthesis along the strand, forming short segments called Okazaki fragments that collectively form the lagging strand. A structure called the clamp loading protein associates with a DNA polymerase molecule on both of the parental strands of DNA. The clamp loading protein is also bound to helicase. These associations made by the clamp loading protein coordinate the simultaneous synthesis of both DNA strands at the fork. Note that the two strands of a double helix run in opposite directions. They are anti-parallel. While the leading strand can be continuously synthesized in the direction of the replication fork movement, the lagging strand template must be folded so that its polymerase can create DNA in the 5' to 3' direction and still move in the same direction as the overall movement of the fork. Using the 3' end of a primer, DNA polymerase III lays down new DNA in the 5' to 3' direction until it reaches the previously synthesized Okazaki fragment. The DNA polymerase then dissociates from the DNA, but remains attached to the clamp loading protein. The clamp loading protein adds a new clamp and places the DNA polymerase at the next primer. The process repeats. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.
Oh, I'm so sorry. And that's the end of this video that overviews the DNA replication. Okay. Do people have questions? No? Okay, I'm going to continue. If it lets me go to the next slide. Okay, so in DNA replication, you have the origin of replication. And here is where the cells will begin replicating their DNA at this replication origin area during S phase. And cells can have more than one origin of replication. And in human DNA, there's billions of base pairs. So this is why they can have multiple origins of replication. And the origins don't fire at the same time during S phase. And just like the video said, replication occurs bidirectionally. So in two directions. And does anyone have any questions at this point? Okay, continuing. And so the video also talked about the replication fork and how the origins of replications fire off in both directions. And helicase, the enzyme helicase, unwinds and unzips the DNA during DNA replication. And this is just a diagram that sort of displays what the video was chatting about. And this image here just sort of explains most of the proteins and major players, it says, in DNA replication. So you see that you have helicase, which unwinds the strands. Then you have the sliding clamp, which prevents DNA polymerase from falling off the DNA. Okazaki fragments, RNA primer, the two strands, leading strand, lagging strand. And there's also sliding clamp loader, which is not pictured on there, but that's an ATPase that uses energy to open and close in order to load the sliding clamp on and off of the DNA. So yeah, this is just the image that just describes what we spoke about or what the video mentioned. So anybody have questions? Okay, I'm gonna continue. Sorry. And yeah, this slide just gives a summary in written words instead of a picture of the major proteins that are involved in the replication fork. And I don't feel a need to repeat myself because I felt like I explained most of these in here. Only one I think I didn't explain is single-stranded DNA binding protein, which is always bound to the single-stranded DNA after the DNA gets separated. And so it just sort of stabilizes that single-stranded DNA. That's why it's there. And yeah, I think I'm gonna go ahead and continue if there are no questions. And the video mentioned leading and lagging strands. I think there's more on this slide. And the polymerizing of a new strand is always from the 5' to 3' direction. And directionality is critical because it determines whether you're leading or lagging. And if you're synthesizing 5' to 3' in the direction of the fork, you're leading. And if you're synthesizing 5' to 3' in the opposite direction, you're a lagging strand. So does anyone have questions?
I have a quick question. Could you explain a little bit more about the synthesis and how on like either side, how like you determine which side of the origin is gonna have a lagging strand or a leading strand?
Sure. May, did you want to explain this question?
Sure. Okay, so I feel like okay, there we go. Are the lines showing up on this, or is that a different?
What do you mean arrow?
Okay. Anyway, sorry, I can't really see a bunch of things. So basically, if you're at the origin, if you were to like start looking at the direction of the fork and then compare it, let's start with the top. See how like on the right it says 5'. So like if you want to make something 5' to 3', like if you know that the thing complementary to the top strand needs to be 5' to 3', like in the opposite direction, if you follow that direction of the fork, it's going to naturally be in 5' to 3'. The natural opposite is going to be going to make it the leading strand because it's going to be 5' to 3'. But if you look at the direction of the fork compared to the bottom, if you look that the 3' is marked, the 3' side, that means if you were going to in that same direction, like, and you're synthesizing 5 prime to 3 prime, that means like the 3 prime would kind of like would be in the same way, I guess. So you'd want to make sure that that one would have the fragments to make sure it goes backwards. It's kind of hard without drawing it. Did that sort of make sense? Or I apologize. I know like that was probably
Yeah,
not.
I mean, it kind of makes sense. Like, I understand like the process that like on the top, I know that it's going to be like the top strand is going to have a leading strand, and then the bottom one is going to have a lagging strand if we're looking at the one on the right.
Here, wait. Let me try to share my video. I'm sorry. I
Kind of
just got back from work because someone like couldn't take my shift.
May, do you want me to stop sharing the screen?
Yeah,
Okay, sure.
do you mind? Okay. Can people see what I'm doing?
Yep, me too.
Okay. So, let's say you have these are your strands, right? And here's the bubble. Can everyone see that?
Yep.
Yeah. When
Okay.
you hold it up close, it's visible.
Okay, yeah, okay. So then, if this is your origin, let me get like a marker. Okay. So, here's your origin. This is going to be the direction that you're going in. And let's say the top strand is 5 prime, and this is 3 prime. So naturally, if you want something to be synthesized 5 prime to 3 prime, that is going to be assuming that you're starting 5 prime, the arrow, let's say the arrow, like this is the directionality of the arrow. That's like what you want, 5 prime to 3 prime. So if you compare it to the top strand, like these two, that's naturally anti-parallel. Like naturally, 5 prime is over here, and 3 prime is going to end up below it, and 3 prime is going to end up here above it. So that is what you want. However, since that directionality is the same, like 5 prime to 3 prime, keep that. But then if you compare it to the bottom, see how 3 prime is still there? That means you need to avoid 2 3 primes right there. So that's how you'll know that this one is going to be the fragments. Does that sort of help?
Yeah, that makes sense.
And it would be the opposite like on the other side. So like if this one is the 5 prime side here, then the 3 prime would be here because that's 5 prime to 3 prime. And then this assuming you're making it 5 prime to 3 prime, and that's when it would switch. This one would be the fragments because you're going in the opposite direction. But so always assume the directionality of the fork is 5 prime to 3 prime, and then you compare it to the top and the bottom to see which one is going to line up opposite and different. And if like the directionality is the exact same as the bottom one, that one is going to be your lagging strand because you don't want to have them the same ends like on top of each other. Does that help? Like, I'm sorry for this like mess, but
No, yeah, that helps. Like drawing the boxes around the directionality and like showing the anti-parallel, I just think is helpful.
Okay, cool. I can try to put something together. I'm not like to make it a little more clear and send it out after. But yeah, that's basically your small thought process. Do you have anything else to add, Michelle?
Not really. I feel like you touched upon it pretty well.
Okay. This was honestly like the hardest. I took genetics, and not gonna lie, I did not understand any of this when I took genetics, so it takes a while.
Same here. I feel like it's difficult to understand the first time you're learning it. But yeah, so that concludes leading and lagging strands. Hold on. Oh, May, would you like to do the next slide?
Sure. Yeah, so I guess this is kind of what I touched on, just like a little more in depth. So when you have your lagging strand, you want to synthesize in the opposite direction. It's going to be, that's when you're going to make your Okazaki fragments, since it's not continuously synthesized, because it's that sort of thing where you need to make sure that it ends up being 5' to 3', but it's not going to be as smooth. So each fragment is going to require a primer for DNA polymerase to bind to it, and whenever DNA polymerase jumps to it, the sliding clamp is needed as well. So yeah, kind of think of it as something like binding, then releasing, then binding in a different spot, then releasing. It's kind of, yeah, and it only latches on for a certain amount of time. So yeah, when all of the DNA is synthesized, the RNase is going to remove the primers, and DNA polymerase is going to go back to fill in these spots with nucleotides. And that's when you have the nicks are going to be sealed by DNA ligase from it. So it makes it continuous again.
Do people have any questions on that? I'm gonna assume no, and I'm going to continue. Oh my goodness, I was worried it was gonna start playing. But anyways, these are more videos I just put in the presentation about DNA replication. I know it's a tough concept, so I just looked up videos on YouTube that I thought would explain the concept pretty well. And I think the first video we watched was pretty good, but I always think it's good to just have just a couple of different ways that it's explained. So yeah, so definitely look at these two videos or anything else out there on YouTube or Khan Academy to help. And now we're going to discuss DNA repair pathways, which is the content we did this week. So DNA structure, this is just to recap. DNA, you have your 4 bases, right? Your adenine, your thymine, cytosine, and guanine. And A pairs with T, C pairs with G, and these base pairs occur through hydrogen bonding, and they're phosphodiester bonds that link nucleotides together within a single strand. And DNA structure is a double helix, so sort of like a twisted ladder. So that occurs when the base pairing of 2 strands happen together. And DNA as a single strand can also form a helix somehow. And the strands of a double helix, I believe, face each other in opposite complementarity. So one is in the 5' to 3' direction, and the other one is in the 3' to 5' direction. And this is called antiparallel. And the 5' end has a 5' carbon attached to just phosphate, whereas the 3' end is an end with a 3' carbon attached to a hydroxyl and nothing else. Do people have questions on the DNA structure? No? Okay. May, do you want to do this slide?
Sure. So we're going to talk about mutation rates. This can be a good thing in the cell because they're definitely normal. It's how species evolve. Species will divide a lot and have higher mutation rates and can afford to lose offspring sometimes if they have a lot. Others, like it's just like an evolution thing. Species that don't divide often, like humans, have relatively low mutation rates and are tightly regulated with DNA repair mechanisms. So the kinds of DNA damage that we'll see is single base changes, so mismatch, not really damage, more of a mistake, depurination, deamination, and pyrimidine dimers. And we'll also see some double strand breaks. So keep in mind, like if a double strand break, like DNA is already pretty stable. So like if the double strand is breaking, that's serious. And then there are different repair mechanisms for each of these things that will target the damage in the DNA and try to fix it. We have our single base changes, which is mismatch repair, base excision repair, nucleotide repair, and our double strand breaks, non-homologous end joining, and homology directed repair. It's a mouthful, but yeah, we'll be talking about those later.
Yeah, and I just wanted to make a note here that Professor Francis will, instead of saying the mouthful of non-homologous end joining, she just says NHEJ. And then I think she does the same for homology-directed repair. Maybe she says HDR. But yeah, that's just a side note. So does anyone have questions on this slide? Okay. So this is just a slide about DNA damage and endogenous reactive oxygen species from metabolism and exogenous sources, UV energy particles can cause DNA damage. And there's depurination, which is when oxygen breaks the bond between sugars and a base, essentially, which means that a nucleotide loses its base. There's deamination, when oxygen attacks carbon in cytosine and changes the functional group from an amino to a carbonyl group. And this causes the cytosine to change to uracil. And then another type of DNA damage is pyrimidine. Sorry, I struggle with the word pyrimidine. I think it's just saying pyrimidine dimers, which is high energy particles that are connected through covalent bonds between these pyrimidines, such as cytosines and thymines, that are aggregated together, and it makes a distortion in the DNA backbone structure. And we'll see this when we cover that type of DNA damage and how the mechanism works to fix it. And yeah, so that's DNA damage. So does anyone have any questions? Okay. And I wanted to briefly go over the vocabulary because there's some words that sound sort of similar, but I just figured it'd be helpful for you guys to have them defined. So an endonuclease is a nuclease that cleaves the phosphodiester bond between nucleotides anywhere within the DNA sequence. And the way that you can remember what this means is that the prefix endo means within. And we've seen the prefix endo before in this class when we talked about the endomembrane system during the protein trafficking unit, which I think was around week 3, week 4, possibly. But anyway, so when we spoke about the endo in the context of the endomembrane system, it's the system that's within cells that transports proteins. And also you can think of the word endoplasmic reticulum because it's an organelle that's inside a cytoplasmic membrane and has its own membrane, so within itself. So like endo within, and then I believe plasmic has something to do with the word membrane. So it's just a membranous organelle, then the ER. And so exonucleases are nucleases that remove nucleotide from the ends of DNA. So those can be exposed 3' or 5' ends. And a way to remember exonucleases, which I think might be easier than the endonucleases example, is just like exonuclease sounds a bit like exit, the prefix exo. So an exit is just the back of the building or the very last door towards the back of a building or an airplane. And so you can just sort of think of that as exonucleases remove nucleotides, from the ends of DNA. And the last vocab word I have is ligase, and ligases create phosphodiester bonds between exposed 5' and 3' ends of DNA. That DNA polymerase cannot create bonds between without adding a nucleotide. And ligases just come from the Latin word that means to bind. And I just wanted to reiterate that most of the time in this class, if a word ends in ase, then it's an enzyme. So ligase is an enzyme and a protein. So does anyone have any questions about the vocab? No? Okay. So now we're going to discuss mismatch repair. But before we do that, I have a video on the mismatch repair that I hope the sound works for. So let's look at that.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the mutase protein. Mutase binds to a mismatch site and forms a complex with another protein, mut-L. The formation of this complex recruits mut-H to the region. Mut-H binds to a hemimethylated site. The mut-S, mut-L complex interacts with mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand, which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
All right, so that's the end of the video.
Although rare,
And May, do you want to explain this slide?
Yeah, so kind of as the video touched on, mismatch repair, this is going to happen after all the proofreading happens, because even with DNA polymerase proofreading, there's like still errors, not as many, but there still needs to be ways to catch the mismatches. So a mismatch is when a mispaired nucleotide is added to the synthesized strand. So this can distort the backbone. So as you can see in the diagram, in A, an adenine was added when it should be a cytosine, and it's across from a G, a guanine, and those don't match. So this can be an issue if the mistake never gets fixed and is translated like further on. So as you can see, if there's no repair, the next round of DNA replication, there are two different like ways of targeting. You can either have or not targeting. There's two different outcomes you could have. You could have like the mutation stays where an A like is the one that is following and a T is now going to be inserted, or like the A is going to be removed and a C is going to bring it back to how it's supposed to be. It goes more in depth on
The next slide.
the next slide. It's probably a better visual. Yeah. So yeah, in order to make sure that the cell fixes the mistake, it needs to recognize which strand. So mismatch proteins are going to recognize the mistake and which strand is which. So yeah, as I was saying, like if you repair the old strand, as you can see like on the left side, or yeah, only the template. So then in the next round of DNA replication, the mutant is going to stay. But if you only target the synthesized strand, then it's going to go back to C's after that. And how are the strands different from each other? Prokaryotes are going to have methylation of adenines in a palindromic sequences, and eukaryotes are going to have NICs in the newly synthesized strand. But once all the NICs are sealed or the DNA is methylated, MMR cannot occur. There's like a small window of time after the replication where this is occurring. Yeah, so for this, like the main idea is that when a mismatched, like mispaired nucleotide is inserted, there are two different ways that it can be targeted to repair, and that will change the outcome of what is continued to be synthesized in further replications.
I'm not sure if you guys know this question, but I know that she like talks about how like there can be mutations if like they don't correct the right strand. How often does something like that occur? Like how often will mismatch repair occur properly? Or is she just using that as an example? Like if they like yeah, I guess is she just using the like ones that are wrong as an example, or does mismatch repair have errors too?
Well, I have no idea. I can look into that and like ask her. Michelle, do you have?
No, I'm also not really too sure. But that's definitely something we can ask her. Sorry, I wish I knew.
Okay, no, it's just I was just curious because I know she like mentioned it, but like it was never mentioned whether or not like those mutations like are something that occur, or whether or not it was just an example of like what would happen if they didn't recognize the right strand.
Okay. Well, we will look into that and get back to you on it. But as of now, not too sure. Okay. So do people have any questions on this slide upside besides from that question? No? I'm gonna continue. But May, why don't you just do this slide as well, and then I'll just do the next mechanism of repair. If that's okay.
Yeah, that's fine. Yeah, so this is, we're going to focus on the prokaryote mechanism for MMR. So your step number 1 is MMR proteins are going to recognize the mismatch. So the different nucleotides and other proteins are going to, and they're going to determine which strand is the newly synthesized strand for proper correction. Next, an endonuclease is going to create nicks in the DNA. And helicase is going to unwind the DNA, so it will kind of cause the nick strand to peel, sort of, and then the exonuclease is going to chew back on this like peeled strand, so it excises mismatch and exposes the single-stranded DNA. And then the single-stranded DNA binding proteins are going to bind to the exposed strand, and DNA polymerase with proofreading is going to resynthesize the strand, and then a ligase will seal the nicks, like as we've seen in other like things from the newly synthesized section. And then you have your methylase, which is going to methylate the newly synthesized strands. It is similar to the mechanism of eukaryotes, but they don't necessarily need the endonuclease if nicks are present. So yeah, that's kind of it. It's nice. It follows with the diagram, basically.
Yeah, so it's just sort of re-emphasizing what May said on the last slide. So does anybody have questions on this slide? Okay, let's continue. Okay, so nucleotide excision repair. Before I get into nucleotide excision repair, here's another video.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix.
This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
All right, so that is the video. And I just wanted to say that I believe the UVRC is the endonuclease, and then UVRD is the helicase. But I'm going to get more into nucleotides.
Michelle, we can't really hear you.
You can't hear me?
It's just really quiet. I just wonder if it's just like this, I think, after you play a video. I don't know why.
Sorry. But I was saying.
Go to the next slide. Like, you'll be louder. I don't know.
Okay. Video. But can people hear me now?
You're still pretty quiet.
Okay. I'm coming out clear on my computer, so when I upload the video, it will be clear. But I'm just gonna try and see if I can talk louder, because I'm not too sure what's happening with the audio. But anyways, so nucleotide excision repair repairs major distortions in DNA, such as those caused by pyrimidine dimers. And like I mentioned in the video, the UVRC in the video was the endonuclease, which cuts on either side of the dimer, and it takes out a chunk of the DNA, so removes about 4 to 8 bases on either side. And then once it removes it, then DNA helicase will come in to unwind the DNA and peel off the next strand. And this removed strand is degraded. And once this removed strand gets degraded, you'll have your single-stranded DNA binding protein, which binds to the exposed strand, because single-stranded DNA is not really stable on its own. So those proteins have to be there to stabilize it while it's being repaired, while the DNA is being repaired. And DNA polymerase and DNA ligase will come in and fill in the gap of the pyrimidine dimer and the DNA around it that's been taken away. So then the DNA polymerase and DNA ligase will just fill in the gap completely with complementary nucleotides, and this seals the NICs. So does anybody have any questions on this slide? No? Okay. And so for this pathway, mutated nucleotide excision repair can result in a disease called xeroderma pigmentosum, which I believe has to do with spots on the skin. Yeah, so I'm not too sure if you have to know in detail about that, but that's just something that I knew. I remembered learning from Bio 151. And mutations in, I think that also includes some information about the other repair pathways. So mutations in mismatch repair proteins can lead to colorectal cancers, endometrial cancers due to microsatellite instability. And microsatellites are tiny repeat regions of DNA that repeat 5 to 50 times. And when these regions of DNA are unstable, the instability is caused by deficient mismatch repair, and that makes these regions susceptible to mutations when they're unstable. And mutations in base excision repair can lead to the loss of viability during embryogenesis, which means that the organism does not develop. So this is just talking about mutations as a whole in the 3 pathways, DNA repair pathways that we're going to talk about today. So does anybody have any questions about this slide? Okay. And I believe the last DNA repair pathway is the base excision repair. And I don't think it will be any surprise to anyone that I found another video. So we're gonna just go ahead and play this video, and I think this is the last video.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick, and the repair process is complete.
All right, so that's the end of the video for base excision repair. And you can see that the video does a good job of highlighting just the difference between base excision and nucleotide repair, because base excision just removes that particular area or that particular base, whereas the nucleotide, I don't know why it's not letting me go back to the slide. Sorry. Whereas the nucleotide excision repair removes the pyrimidine dimer and then some of the bases on either side of it. So some of the bases surrounding it. But anyways, May, do you want to explain this slide?
So basic excision repair, so it specifically repairs deamination and depurination. I don't know if Michelle used this like before I got here, but yeah, basically amination, like that signals, like I don't know if you guys are in Orgo right now, it signals like in amine groups that like if you see like an NH2, that sort of thing, then classifications, you have your purines, pyrimidines, that has to do with like cyclical structures, but just so you know, like background of that. So for that, you have 4 like small steps. Number 1, mutation is going to be recognized. So in deaminations, uracil DNA glycosylase is going to find the uracil and cut out the bond between the sugar and the base. So that's your A basic site. A basic sites occur in depurination as well, and BER is going to be picked up at this point. Second, apurinic / apyrimidinic endonuclease and phosphodiesterase cut bonds on either side of the A basic nucleotide. Number 3, DNA polymerase is going to fill the corrected nucleotide. The clamp isn't needed for this processivity, but it may coordinate like other proteins. And then your ligase, again, is going to come in and seal the nicks between the 3' end of the added nucleotide and the 5' end of the existing strand.
All right. Does anyone have any questions on this slide? Okay. So then next we spoke about double strand repair mechanisms. And I wanted to point out that for these mechanisms, you should have just a general outline, I believe Professor Francis says, for each process. So you don't have to know them to the detail that we covered the other repair mechanisms, which I believe are the single strand. So the way that we covered the mismatch, the base excision repair, nucleotide excision repair, those ones you need to know in pretty good detail, the way we went over them in lecture, but for double strand repair mechanisms, you just need to know the general ideas. So yeah, I'm going to continue. So there are other types of DNA damage. Double strand breaks, DSBSs, can occur in cells as a result of high energy particles that can break covalent bonds. So these are things like, intense UV rays, maybe if you're out in the sun for too long or something, or ionizing radiation that causes DNA polymerase to pause. And another reason that double-stranded breaks occur is because there are off-target effects from DNA endonucleases. And double-stranded breaks can lead to programmed cell death or apoptosis, premature aging, and translocation events, in addition to cancer. And cancer cell hallmarks or karyotypes of cancer cells, and then this occurs when chromosomes are abnormal. And these types of DNA damage, the DSBs, can be repaired through non-homologous end joining and homology-directed repair. And you can see in the image below that you have the chromosomes that a person has look very different, and that is due to the double-stranded breaks. So the first tile or the first chromosomes on the left are like a different in size, shape, and you can see that for some of them on the right side, when they're looking abnormal, that there are extra copies of them, and they're just either different shapes or different colors are highlighted, which I think indicates something about the DNA or genetics within them, but you can see that they look clearly different, and that's most likely due to the double-stranded breaks. So does anybody have any questions regarding this slide? No? Okay. May, do you want to do this slide?
Then we have NHEJ, so non-homologous end joining. So this is used for double-stranded breaks when replication isn't occurring. So you don't need a duplicate DNA strand. So what happens is you have your exonuclease, and it's going to remove the overhang of the jagged ends of the DNA. So you can see like you have your double-stranded break like in the diagram, and they aren't like lining up. So with your exonuclease, it's going to make a clean cut in the double-stranded RNA, so they're like more even. And you're going to lose nucleotides when you do that, since they went from jagged to like being lined up. Sometimes they can add nucleotides, but they aren't always the right ones. So then again, you have your ligase that's going to join the DNA together. So yeah, it's not the most ideal mechanism because it's kind of like simple, I guess. But that's basically it. You're combining your double-stranded DNA strands after you cut the nucleotides out.
All right, does anyone have any questions on NHEJ? Okay. And I believe the last DNA repair mechanism we looked at, or the one for the double-stranded breaks, the second one for double-stranded breaks, sorry, is the homology-directed repair, homologous recombination, which it's also called. So this happens when you have double-stranded breaks and a duplicate copy of your DNA during cell division. And the way it works is that it uses the good copy of DNA to fill in the gaps. And this method is just more effective at repairing double-stranded breaks compared to the NHEJ, just because it uses a good copy of your DNA to fill in the gaps, and the process just is more, I think it's just better off than when you have a broken double-stranded DNA and it's accidentally broken and the endonuclease just breaks it even more. I just think that this method is more effective for the cell. So if the cell could, it would probably use this method every time it had a double-stranded break. And so you can see the diagram shows you that you have the double-stranded break, and then you also have the duplicate copy of DNA, which happens during cell division. And then the nucleases will digest the 5' ends. And then there's homologous recombination, which is just like when the strands sort of mix with other strands. And then that ends up causing new strands to be produced where the double-stranded break is fixed, and then it ends up being ligated together through DNA ligation. And yeah, that's homology-directed repair. Does anybody have any questions on this? No? Okay. I actually think that's the last slide for today. So that is everything for this review.